task. We could use the hour that we normally would have in class from 12 to 1 and then just have a review of what you need to know, or we could have it on a different day next week. Do you guys have a preference for which one you'd like to do for your review? So immediately after class, it will be about 30 minutes. I'll just cover the slides that you need to make sure that you know, and then also answer any questions. Hopefully you'll have studied by then. So if that's okay for everyone, we will have sort of a 2.5-hour class. The half hour will be a review to help you get ready for the upcoming term tests. And that will be like the one time that we have to do that. Is that okay for everyone? Sure. All right, thank you. And are we okay here?
We think so. We're just checking. Is the audio recorded? The local one? That one?
Yep, I will. So the mic is good.
Yeah, everything's. Oh, sorry. Here you go.
Okay, send projector. Oh, that's terrible.
Oh, did it not?
Still don't see it up there though. It's not going up on the projector though.
I think when this finishes.
No, well, when it goes back to the startup screen.
Oh no, that's fun. Is there a way to like turn it off and turn it on again?
I think it's already done it twice. So if it doesn't work this time, we'll just kind of go with whatever.
Okay. Please.
Let me try one other thing.
Yeah.
Just in case. Try flipping. Okay, send this to.
Oh, no, project.
So that works. Then this should work too. Yep, and I think it does.
Yes.
Okay. Okay, good. So I think we're good.
And that's also
Yeah.
good. So.
Okay, awesome. Thank you for your help. Okay, so again, just to get you ready, because I'd like you all to do really well on the upcoming midterm. It'll be 60 minutes long, 35 marks in total. It'll be very similar to what I posted for you as a term test from last year, so it won't be very much different. The short answers you can go through and sort of check that out. Section C will be a little bit different this year, where you'll have to either describe something or design something, but in next week's review session immediately after class, we'll take up a couple of possible questions so that you have some better idea of like what to expect, and I promise it will be a completely fair term test. It'll be the same format, it'll be a very fair final exam as well. There's a lot of material that we need to get through. You all have different backgrounds. Some of you are more neuroscience related or oriented. Some of you are more genomics related. So I'm going to try to make sure that we all sort of have a similar background, understanding that we may not have as much detail on some aspects as we do on others. So right now we're looking at one of the hottest techniques. We don't actually know if this technique will continue to dominate cell molecular biology. There's already some hints that CRISPR-Cas9 is maybe not going to be around after about 10 or 12 years. It's been a really interesting sort of technology that was first put forward by Jennifer Doudna as well as Emmanuel Charpentier. And of course, many of you will know that there is a lot of controversy with Fang Zhang and MIT. So there are two different groups that are looking to win the patent war on CRISPR-Cas9 technology, but we're actually not 100% sure if this will be the dominant technology. It is right now, And it's a really exciting technique because it allows us to take the genome and to modify the genome, but we think that there are other better techniques that are actually starting to appear. Again, just because some of you are asking, and I need to make sure that you're all aware of what you need to do. So when we're looking at participation, you are going to give some feedback. That will be the peer evaluations. That will occur much later on. You are going to have to pick a paper. You're going to have to come in and see me. And so you will get a mark based on the paper you've chosen, how you've decided to break up the assignment, and you'll get a mark at a 5%. Usually it's a really good mark as long as you prepare well for it, and hoping that your group will be no different. In the term test, you will actually be rewriting the same term test again as a group. You'll be in one exam center. If you happen to be writing in accessibility services or have an accommodation, there is something else for you to do as well, so you don't have to worry that you'll be missing out on any of these different things. So just to get you aware, in the next 2 weeks, part of your participation marks will actually be going up. So the post-midterm assignment, that will be sort of your 5%. You'll get your mark on that immediately. You'll get a mark based on what you do as a group. If you get 100% on your term test as a group, that means that you'll get 5% out of 5%. And so picking a good group is really important. What do you think the average is on the group midterm? You must have a guess. I mean, pretend it's a multiple choice test question. You have to guess on something. Yes. 83.5. Nice guess. Actually 83.51, but that's pretty close. Yes, did you have another guess? Maybe 95. It's usually in the high A's to A pluses. So I'm assuming that as long as everyone in your group has studied, you'll all do well. And it's your opportunity to kind of discuss as a group some of the same questions again. So that means that you'll be able to go and review that material twice. So today we're going to look at targeted genome editing. And again, I'll kind of point out a few slides to you as we go along. We think it's really important for a variety of reasons. Not only can we potentially, we haven't done this yet, there's one clinical trial going on with RNAi technology, which is actually a really old technology now that is working on therapeutic intervention. But if, for example, CRISPR-Cas9 maintains sort of the way in which it is being developed, it's possible that we might be able to actually alter our genomes. Again, it's a little bit of a stretch right now. No one's even come close. The closest we've got so far are some primates as well as some larger mammals like the dogs that I mentioned earlier in last week's lecture. But we also know that CRISPR-Cas9 is working currently for gene drives, and so pathogenic organisms, their genomes are being altered, or other types of genomes are being altered, and that is directly making an impact right now. And so we think that this targeted genome editing, being able to not only change our genomes, but potentially pathogenic genomes around us, might be really important. So that means that we need to understand that technology. We need to have sort of a common base. So we're going to start off with is that model that we looked at last week, which is muscular dystrophy. So these are those inherited diseases, primarily of skeletal muscle. Can also involve cardiomyopathy, so it's not just skeletal muscle, but other muscle types that can be affected as well, like cardiac muscle. And so because it's affecting a number of different systems, and because it's progressive, once you have it, and once you pass a certain developmental stage, and you're no longer making dystrophin, you are not going to have proper muscle functioning, and muscle function gets lost, and it often leads to the condition being fatal. We know that there are genetic models, and we know that there are a number of genetic mutations that involve DMD or Duchenne's muscular dystrophy. And we know that we have some model organisms that we discussed in the last lecture, which, by the way, you need to know for your upcoming term test. That was part of your study guide, and you need to make sure that you know all the different details of the different model systems that we talked about, whether it's the CRISPR-Cas9 rat model that I mentioned briefly, the different dog models that we'll again expand on next week, and then the MDX and NDX52 mice that I mentioned to you last week, and today's model, which is zebrafish, which I'll give you an overview of today. So muscle itself, if we take a look at it, have a number of different inheritable diseases, not just muscular dystrophy. There are a number of different types of skeletal muscle diseases. These can also be, for example, acquired. So you can, through infection, actually damage skeletal muscle or cardiac muscle. And as a result, we need to be aware of some of the molecular pathways that might actually occur. And for example, a cardiomyopathy, if you have lost oxygen to cardiac tissue, might actually lead to abnormal growth later on and scarring of that cardiac tissue, leading to a dysfunction in normal heart function. So when we're looking at the overall way in which skeletal muscle and cardiac muscle tends to work, we need to understand a lot of the basic cellular pathologies that are involved in this pathway and whether or not we can alter them. Some of the acquired cardiomyopathies and skeletal muscle pathologies, they're not quite at the stage where we can take an acquired. So you have had an infection, and as a result of that infection, some of the pathways are affected. We're not quite at the stage where we can go in and manipulate those acute systems or acquired systems, but still understanding the pathology, the cellular molecular basis for those is actually helpful, at least in the developmental diseases like Duchenne's muscular dystrophy. And especially as we understand the genetics, why does one gene being altered affect a molecular process that then affects how that skeletal muscle cell functions is really important. One of the best model systems that we're currently looking at right now is actually the zebrafish. Some of you might be doing research related to zebrafish because zebrafish models are increasingly becoming important. The technical name or the scientific name for zebrafish is Danio-Rerio, and so you'll often hear about this in some of your second year courses, and I just wanted to go back and review a little bit on that. To be honest, I've worked with almost every single animal model, so I've even worked with primate model systems previously, so I analyzed their brains at Toronto Western a long time ago, and I've worked with C. elegans, I've even worked with Drosophila, and I've actually worked with Xenopus laevis, but I haven't actually worked with zebrafish. So at some point, I'd really like to be able to do this because it's such a powerful tool in terms of developmental molecular genetics and understanding these different pathways. So again, here's an example of what I mentioned to you in the last class. So here are both bullet points as well as a diagram. And on this diagram, you don't really have to worry about the number of hours that occurs between fertilization and some of the events that I'm going to mention, but there are some key points to why Danio-Rerio or zebrafish is actually really important. So these are diploid embryos, and we know that these are vertebrate embryos. So they're more like us. So C. elegans, great for genetics. We understand and know every single cell type. We know all the genes that are involved in C. elegans, but it's an invertebrate model. Zebrafish is a vertebrate just like us. It has a nervous system that looks a lot like us. It has a muscular system, even though it's a fish, the body plan is very similar to our body plan. And because of the way in which it can actually develop very, very quickly from the time it is fertilized to the time that we can start to see individual organs or different organ systems or organogenesis, and before it actually hatches, we can actually track each of these different stages. And it occurs very, very quickly. So it's a big advantage to us. It also allows us, because we can track each of these different stages from the time that the eggs are fertilized all the way to organogenesis, because we can track in terms of the number of hours in between these different stages. So each of these would correspond. The number of hours or the hour that you see here is not important. Because of the way in which we can track each of these developmental stages from fertilization to organogenesis, and you don't need to worry about the individual stages that you see here, we can actually use these for mutagenesis screens or mutagenic screens. So we add a compound and we can see at different stages when we add it, we can actually see that it results in a disruption in this organ 15 hours later or 3 hours later. And so it becomes a really important system. In addition, as soon as, or very, very close to the time that it hatches, its muscular system is already fairly well developed. It starts swimming. So unlike us, we don't actually move around all that much. We know that the zebrafish can actually engage its skeletal muscles fairly early on. Again, the other thing that makes it useful for us in the context of Duchenne's muscular dystrophy is that we can look at a number of different muscular dystrophies, a disruption in muscle function, that can be accounted for in a very similar way to what we saw last week. So just like human muscular dystrophy, we can also see that if we were to disrupt this dystrophin complex, and this is known as the dystrophin-associated protein complex, not just dystrophin, but dystroglycan, all of the extracellular matrix molecules, that in fact it looks very much like the type of system that we saw in the human model system. So this is an interesting model, and this model allows us to study Duchenne's muscular dystrophy. So again, this is an example of a slide that has both bullet points as well as having a diagram on it. The diagram you do not have to memorize. You don't have to actually go out and draw this again. It's really just to orient a couple of the bullet points and some of the important things that I want to share with you on this particular slide. We started looking at, in the last lecture, the genetic structure for dystrophin. Why is dystrophin and the loss of dystrophin so important? Again, if we were to take a look at the overall structure of the protein, which is shown to you here, there are a number of very distinct domains. Obviously an N-terminal domain. We have a number of regions here in this raw domain where we will, for example, have many repeating units, and these are all identical repeating units, and these look a lot like spectrin. And again, we have a C-terminal domain, etc. So because there are a number of identical repeating domains, losing a couple of those domains may actually not be so important for us. So keeping that in mind. And this is sort of the molecule dystrophin that is eventually going to form this complex that we see here. So this dystrophin-associated protein complex includes everything that's here on this diagram. And again, I don't need you to understand all of it or memorize all of it. It's really not the point of going through Duchenne's muscular dystrophy. But if we were to take a look, as we would expect, there has to be a link between dystrophin and one of the major contractile molecules, actin. So this is the filamentous or F-actin, and it links the skeletal muscle. And this would be the part that would be involved in the actin-myosin cross-bridge formation, and it has to link somehow to the sarcolemma or the plasma membrane of the muscle tissue itself. On the other side of things, toward the C-terminal, we know that it's going to interact with dystroglycans. We saw that already, and it does so through a number of other accessory molecules that are shown for you here. And again, I'm not going to ask you to memorize the entire complex. I think it's a really interesting thing if you're involved in its research, but I'm not really a huge fan of just memorizing things for the sake of getting you to memorize them. I'd rather you understand why this might be a problem in Duchenne's muscular dystrophy. I am going to mention that there is a domain on here that isn't really well described, where this will also be involved in coordinating the activity of nNOS, or nitric oxide synthase, as well. And I'll get back to that a little bit later because it becomes important in a future lecture. One other thing to make note of, this dystrophin-associated protein complex that we're looking at is the mature version. We actually have early on in development, we have utrophin, and some of you were asking, so if utrophin can actually take the place of dystrophin, why do we have these different issues? Eventually, we stop producing utrophin. It's sort of a developmental isoform, And it can take the place, it's distributed in the same tissues, but once we need to turn on dystrophin, because of the way that it interacts with a number of other complexes, it will eventually disappear. So it is a developmentally regulated isoform, utrophin is, but the mature form is that dystrophin that we are looking at in this particular diagram. So we had already looked at in the last lecture about dystrophin. We looked at how it plays an important role in Duchenne's muscular dystrophy because it's not just found in skeletal muscle. We also have associated with Duchenne's muscular dystrophy, we also have cardiac muscle tissue being affected. So these individuals not just have muscle wasting, but also have cardiomyopathies. Their heart muscle isn't working well. And keeping this in mind as well, there's also a function where dystrophin, where it's normally interacting with a number of muscle proteins and the extracellular matrix, loss of dystrophin also affects cognitive abilities. And you need to think a little bit about that because in the past, we often have ignored that Duchenne's muscular dystrophy involves changes in cognitive function as well. So I'll come back to that. It's still not entirely known like why that's a huge role or why that plays a role, but there are some theories that we'll discuss in the next lecture as well. So we know that in Duchenne's muscular dystrophy, by losing this dystrophin acts kind of like a shock absorber. So for those of you who have cars and those of you who drive around a lot on the Toronto streets, you know that you can drive perfectly well without a shock absorber until your car hits like a bump. Then it's not so pleasant and it can damage your car, for example, if your car isn't able to absorb that shock. And dystrophin is sort of that link between the road and your car, that shock absorber, and we need that. Without it, every time your muscle contracts, the sarcolemma can tear, the muscle tissue itself can be damaged. So we need that dystrophin. It's there sort of for a variety of reasons. So going back to muscle development and looking at the zebrafish model, there are some other advantages as well. And again, you need to understand, especially for potentially a short answer or part of one of those one-line responses, all of these different model systems, we have them and we use them, and sometimes we're experts on them, but we have them because they give us different types of advantages or disadvantages. So we know, for example, that the embryo itself develops externally outside of the mother. So the zebrafish lays its eggs, they get fertilized externally, and we can watch the developing embryo undergo all these changes. That occurs outside of the body, which is useful for us. So development externally is important. The animal itself, a vertebrate animal, is also transparent, so we can actually see the beating of the heart, the contraction of the muscles. We can see all of these different organ systems as they're developing. So also really important for us as well. We don't need special techniques. All we need is a microscope that transmits light. And then there's somatic muscles, the ones that will eventually form muscles like in your arm or your legs, the ones that are involved in skeletal muscle development are actually a very large proportion of the body, and they can start using them very, very soon after gastrulation or after the formation of the different tissues. So a number of these different embryological developmental studies and genetic studies have used that to their advantage. We can see into the animal. It's a transparent organism. That helps to identify different organ development, as well as looking at muscle development, which for us, when we're looking at Duchenne's muscular dystrophy, is also really important. Zebrafish is also one of the first organisms where genome editing actually occurred. So I'm going to, again, use that sort of as a way to introduce some of these different techniques. One of the earliest genome modifying technologies is known as these morpholinos. And these morpholinos are a short form for morpholino oligonucleotides. They are things that we can synthesize, and then we were able to do this a long time ago, back in the late 1980s, early 1990s. And in this way, we can actually knock down the function of a gene. So if we suspect Duchenne's muscular dystrophy involves dystrophin, and I wanted to test my hypothesis that dystrophin is a molecule that's important in muscle development, and I use this antisense technology by making a morpholino oligonucleotide, applying it to the zebrafish, I'd be able to knock down the function of that particular gene. So if I have a hypothesis or if I have a target gene, this was a very powerful way in which to be able to do so. And in fact, morpholino oligonucleotides aren't quite as broadly used as they used to be back in the 80s and 90s, but they're still in use today, especially in drug discovery, especially in molecule discovery, and for large-scale screening to look at the function of different candidate genes. It can also be used to identify potential mutant phenotypes. If I think, for example, a mutation or a small change in a region of dystrophin might lead to a different phenotype where it's not swimming as well, I could use morpholino oligos to do this much more quickly than I could with CRISPR-Cas9. So again, these morpholino oligos are very, very useful. They can do this both in the mother or the maternal animal, as well as the zygote or the fertilized egg outside of the mother to examine gene function. So it has a number of powerful techniques and technologies. You guys have all taken organic chemistry? Were you good at chemistry, organic chemistry? Not so good. Okay, neither was I. It's okay. We'll all survive. You don't need to worry about being able to draw out the structure. If you like this structure, you happen to think it's a really pretty structure, you want to get a tattoo of this structure, feel free. I don't need you to worry that you're going to have to draw this structure out. Some of this structure is on this diagram as well, or on this slide as well as the next slide, especially when we talk about the way in which these phosphate groups are organized. But these morpholino oligos are actually really powerful ways for us to knock down gene function. How do they knock down gene function? There are two different ways in which they do so. One, they can actually block the spliceosome itself. So that's one method. So that's the second last bullet point that you see on here, that we can block these mRNAs, and we can block these spliceosomes, and we can block them at the level of the pre-mRNA from actually being formed. So that's one way in which these oligos do it. So they sit there with the RNA, and they block the enzymes, or they block the different complexes from forming. They use steric hindrance. They're just really big molecules. They're big, bulky molecules, and they prevent sort of the normal complexes from forming. It's exactly the same thing through the other mechanism that the morpholino oligos have been used to block gene function. So we block the mRNAs from being processed, the spliceosome. Another way is just to prevent them from actually being transcribed to begin with, or this being translated to begin with. And so by blocking the ribosomes from interacting, by having these morpholino oligos targeting the AUG sequence, and the untranslated region, we can actually block ribosome assembly. So the RNA might be there, but the RNA that has these morpholino oligos attached to them will simply not be translated, and not having translation is just the same as blocking the genetic sequence. So again, this is a really busy slide. None of these diagrams that you see here on the left are ones that you need to memorize, just like you don't have to memorize the structure, although again, there's a couple of things that are mentioned here as well. So if you were to take a look, we have this phosphorodiamidate, which was on that previous slide, and those phosphate groups provide this big, bulky steric hindrance that will either block the spliceosome from forming, or it will block this initiation of translation and will block the ribosomes from binding to that particular mRNA. So these are the ways in which, by introducing these big, bulky molecules that contain those phosphate groups, that also have the normal sort of base pairs that we would have with RNA, by incorporating these into the RNA, it prevents them from actually being either spliced or to be translated as we would normally expect. So it's just an elaboration of what was there on that previous slide. This was the first technology. It worked incredibly well. Scientists were really excited because they thought, wow, so we do have the potential to do this in an organism and to block gene function. And so it became a really interesting, very powerful tool that scientists got very, very excited about. So do you have any questions on this or anything that you want to clear up now before I move on? Again, understanding that we only have about 2 weeks before you have a term task. Is there anything at this point that isn't so clear for you that I could clear up? Yes, Alice?
Is the morpholino complementary to the RNA sequence that it's looking?
There are 2 different ways to do it, but generally speaking, if we were looking at spliceosome blocking, we would generate a complementary sequence. Anything else? Yes, Aaron?
Why use the morpholino instead of just like normal RNA or DNA? Like what's special about
The phosphorodiamidate groups are really big and bulky. They may not look like it here, but they're big and bulky enough that they will block other parts of the spliceosome from coming in or from different parts of the 40-as ribosome from binding to the appropriate site. So it's just a way to have the same sort of sequence, but to be able to block sort of the normal initiation of either translation or spliceosome functions. Yes?
Does this mimic the other effects that the mononucleotides will have on the cell, such as the cycles of the CMP, for example?
Will it have other effects on other mononucleotides? As far as we know, I'm not an expert on this because I haven't ever used zebrafish. When different investigators go back and look at whether cyclic AMP or other mononucleotide functions are altered, it doesn't look like it. So it seems to be very specific for these 2 effects. Any other questions? Thanks for a great question, Taha. Anything else? Yes, Emily.
Does it block 100%?
It's pretty close to 100%. So you would actually generate more than one of these, and then you would see which one gives you the best sort of phenotype. Because of the way that these have a very quick life cycle, you'd be able to do a really rapid screen. You wouldn't have to wait like months where you would in like a mouse or a rat. Yes?
Is it reversible, or do we need to keep on like adding more apples into?
So is it reversible? So would you be able to?
After it's blocked, can it be removed and restart the translation again, or that's?
For some cases, yes, people have used it where it can actually no longer be applied. Usually, though, you try to disrupt it at a specific developmental stage. But if you're looking at something that's long-term, for example, that might be related to a neurodegenerative condition or a much longer condition, you could try to no longer apply the morpholino. It's a good question. Okay, some great questions. Thank you for them. So this slide is not one that you need to worry about. Morpholinos are a very powerful technique. They work incredibly well in zebrafish. They do work in other systems as well. They work, for example, in tissue culture. They work well in mice, but they're hard. They're hard to actually design, organize, and to be able to deliver them isn't always so easy. So one of the reasons why people are so excited by genome editing, which is sort of on this timeline here, and why people are so incredibly interested in this CRISPR-Cas9 system is that it's been a work in progress, and people are always looking at how the technologies kind of evolve. And if we were to go all the way back to 1979, this is where one of the first papers that came out that was reported in a Saccharomyces yeast strain was actually able to show that genomes can actually be edited out. It was one of the first studies. And you can see that the relative timeline has been sort of slow and kind of disrupted. It hasn't sort of been a linear progression all the way through from 79 and a slow steady rise. However, if you were to take a look at the first sort of major paper that came out related to the mammalian genome in sort of the early 2000s, a number of different techniques that were sort of all related. So these are nucleases. They are going to break nucleic acids or DNA sequences Whether you're looking at talins or whether you're looking at zinc finger nucleases or the direct descendant CRISPR-Cas9, all of these have sort of taken off really since about the mid-2000s, 2005, 6, etc. And then it's been a real explosion since that one paper came out in 2012, where we've had so many of these other papers come out since then. And every single year, the number of publications related to CRISPR-Cas9 and genome editing has been on the rise. So genome editing is a very important tool. We think it's important not just for understanding developmental processes, but theoretically we're doing this because if we have a really bad disorder like Duchenne's, which affects about 1 in 3,000 to 3,500 individuals, and it's a really, it's a fatal disorder, so why not try to alter that genome? Could we alter that genome? Could we edit that genome out, which is the long-term goal of this particular technology? So going back, looking at some of the ways in which this technology works, they're all kind of related to each other, and I need you to understand some of the different aspects of these different technologies from that particular standpoint. So again, going back, why are we looking at things like zinc finger nucleases, or why are we looking at things like talins? So zinc finger nucleases are here at the bottom, talins here are in the middle, and then the technology that you probably have heard the most about, because that's the year where some of you actually started university, that's where CRISPR-Cas9 became sort of the dominant genome editing technology. If you entered U of T in 2015, this was just sort of starting to make news at that particular time. All of them kind of work in the same way. There are some analogies between all of these different types of nucleases that you see here. So in both cases, or all cases, we are looking at a nuclease. Here is one nuclease for zinc finger nucleases, FOC1. We need to have two nucleases that are going to cut two separate strands of DNA. So here is the 5' 3', and down here at the bottom, the 3' to 5'. You'll see that FOC1, one of the FOC1 enzymes will cut here, the other one cuts over here. The reason why we need two of these is because FOC1 is an enzyme that requires dimerization, so you have to have two of these in order for it to cut effectively. A little bit later, because it's challenging to actually create these zinc finger nucleases, it was done first, by the way, in zebrafish, which again sort of is why we're looking at this. Also in zebrafish, these talins, which are a little bit easier to design, so these modules that you see here that align to the FOC1 to target it to a specific genetic region, these are very hard to design. They're very hard to actually implement, and they are much more challenging and much more expensive. Talins, which came along a little bit later, these tail modules that will allow for FOC1 to bind to a specific DNA region and to cut it, these talin modules are actually easier to design, and so this is sort of the next generation that came up after zinc finger nucleases. Still that same nuclease FOC1. You need two of those to be able to cut a specific DNA sequence. And you might think that CRISPR-Cas9, and the nuclease here is Cas9, you don't use FOC1 anymore, but one of the things that you'll see is there are actually two different nucleases that cut DNA on two different strands. So there is an HNH site and a RUBC site that is part of the Cas9 nuclease. And so it is very much like what we saw earlier. So it may not look like it, we don't have FOC1, but we are indeed cutting two separate strands of DNA, and I'll show you this parallel in a moment. Do you have any questions about this before we go on? Or any other questions about what you need to know? Yes?
So the main difference between talin nucleases and zinc finger nucleases is just that it's easier to design?
The separate modules that will locate the FOC1 enzyme on a specific portion or region of DNA, they're much easier to design using talins than they are the zinc finger modules. And I'll get to that in a moment.
To
Yeah.
the diagram, I saw that there were more modules needed
Yep.
for the tail finger. I mean, tail.
For the tailends, yes, you need more of those modules, but it's because of the difference in how those modules are set up, and I have a slide on that in a few minutes. Any other questions that I can answer for you right now? Okay, so there's a lot of interest in CRISPR, and normally, and again, apologies, I hope that when we take our break in a few moments, after I get through a couple more slides, if you are taking a break, please grab some coffee, grab some tea. There are snacks as well, but if you could, and you could eat them outside, just for a number of allergies, not necessarily in our class, but in other classes that use this room, just to be safe and to make sure that those students are doing okay. Normally, when we kind of eat in class, we would have either cheese or yogurt, and both cheese and yogurt is actually where CRISPR-Cas9 originated, because when you look at cheese and yogurt, we often don't think about it, but bacteria is an incredibly important part in creating yogurt and cheese and that fermentation process that occurs. For that cheese and yogurt to taste good, bacteria that is normally used to generate those actually have to be fairly clean. If they were infected with a virus or a bacteriophage, the cheese wouldn't taste so good. The yogurt would taste off. It wouldn't taste quite as good. And so we know that CRISPR, for example, in the Cas9 system is a defense mechanism, the immune defense mechanism that bacteria have developed against viral infection. And so it's important in the food industry, and it's really where CRISPR is taking off within the food industry right now. So this interest in CRISPR is actually really important because for us in biology, because we think, for example, that one, it's really easy to do. In fact, this year we're going to change things up, so lecture 3 is going to be a little bit different than it has been in the past. We're going to go through and create a guide RNA in class, and then we're going to take a look at the off-target and on-target specificity and why that's important and how easy it is to actually create this. You could spend 5 minutes and technically be able to design a guide RNA that will allow you to correct a function of something or to remove a particular locus on a gene. So we'll talk about that because it's incredibly easy to create these RNA-guided genome editing tools, and we'll talk a little bit more about that in the next lecture as well. There are a lot of advantages over talons as well as advantages over zinc finger nucleases, and we're going to be talking about some of those in the upcoming slides. So just as again, sort of an overview for you, this is a good slide to maybe go back and review. There's nothing specific that I'm going to ask you to know about the Cre-Lox system. So you know, for example, the Cre-recombinase is a DNA cutting enzyme. Any DNA cutting enzyme we are going to identify as a nuclease. We're going to talk a little bit more about these talons in particular in the next few slides, and then how this differs with the CRISPR-Cas9. So just like the zinc finger nucleases and talons have a lot of parallels and look very similar, and even though these look somewhat similar to the Cas9 nuclease system, there are some fundamental differences that I need to cover with you as well. So we'll go through all of that. We'll use this sort of as the last slide. You don't need to memorize details that are on this slide, but it is a good slide to come back to when you're reviewing. And so when you're studying on the study guide, you'll see in the upcoming week that slide 17 is an important one for you to maybe go back and revisit as a review tool, because many of the concepts are summarized for you on this slide, but you don't need to know the Cre-Lox system. Do you have any questions on any of this before we kind of take our first break? Okay, so okay, as we're coming back from break, some of you are asking how the group mark would be working for the term test. So you'd write it individually. We'll mark your test as quickly as we can, post your marks. That will count for like 20 or 25%, whatever you do better on later in the course. But the 5%, for example, as a group, if you decide on all of the answers, you agree on all the answers, you will get 33 out of 35, for example, which was a very common sort of number last year. That would work out to about a 94.3%, which would equal about 4.71 out of 5, and that's what you would get as your participation on the post-midterm test. If, for example, you don't agree, you can actually indicate like which questions you don't agree on, and then you can further subdivide your mark that way. But for the most part, this was sort of the average on that post-midterm assignment, and there were a lot of perfect marks, etc. So please don't worry about it. It'll be okay. If you're not doing it through the class next week, you can also do something similar in the accessibility or through accommodation, so you don't have to worry that you're going to be missing out. Okay, so if we go back, what I'm going to be telling you about in muscle is actually fairly easy. So in muscle tissue, we can do a lot of genome editing, and muscle tissue, because of the way that it's organized, is actually fairly straightforward. Neurons, though, because this is a neurogenomics course, neurons are actually much more complicated and much more difficult to actually do a lot of genome editing as well as targeted delivery. So one of the reasons why the neurons actually have on their synapses, they actually have what is a dense network of postsynaptic density proteins. If we want to target all of this complex, which might, in theory, look a little bit like that dystrophin-associated protein complex, you might think they look the same, but there are so many more proteins. It's like another magnitude higher, so about 10 times more proteins per unit area than we would have at muscle. It makes it really, really difficult and challenging to use targeted genome approaches. Even creating knockout animals has been really challenging. And one of the reasons why knockout animals or knock-in animals in neuroscience and neurogenomics has been so hard is that there's always some kind of compensation. There's so many things going on at the synaptic level that if we knock something out, either it ends up being so useful or so important that the animal dies and we have lethality, but sometimes we have compensation. Just like we see with Duchenne's and dystrophin, utrophin can take its place. This is much more common in the nervous system, and so we need to take that into account as well. And I am going to go through some of the related CRISPR nicases and other sorts of things in the next lecture, as well as taking you through an exercise that you need to understand for your upcoming term test about what is a good on or off target effect, and if you don't have 100%, does that mean that you shouldn't be using it? And we'll have those discussions as we go through a particular guide RNA, how we designed it. I can tell you you're not going to have to do guide RNA designing on your term test, but I want you to be familiar with how to critique a paper, for example, if you choose one that uses CRISPR-Cas9. So CRISPR is the sort of technology that we're the most interested in right now. It's the easiest of all of the technologies to actually use. It was originally adapted because of the food industry. So when they found that certain bacteria that had been in use for a very long period of time in the factory were no longer producing the same type of yogurt, or the yogurt tasted bad or it tasted off, they understood that those bacteria were being infected by either bacteriophages or viruses, and that led to these changes in the quality of food. They also realized that some bacteria were resistant to being infected, and the reason why they were resistant is because they were expressing these guide RNAs that they had previously been infected and almost developed like an antibody response system, and this is how we developed CRISPR. Sometime in the past, bacteria had been infected. They had taken the viral sequence, chopped up the viral sequences, and now had them ready along with the Cas9 nuclease to attack any invading viruses or bacteriophage in the future. So bacteria have this Cas9 nuclease that cuts viral genome sequences, and they also have the RNAs that are left over from a previous bacteriophage infection. So we now use that in mammalian systems to be able to target specific proteins, and we're particularly interested in using this in modifying synaptic activity, developmental changes in neurons, and potentially, if we were ever to understand all of the genes that are involved in things like neurodevelopmental disorders like autism. So again, we'll cover this after the term test. It's a really interesting paper. I have some issues with that particular paper that I'll go through with you. These are my critiques of the paper related to autism and an autism model. Can you use CRISPR to change an autistic animal that is fully grown and fully reverse that? So again, you have to think a little bit about this. If you have an individual who's been identified with autism spectrum disorder, and they're grown up, they're now an adult, and you use CRISPR-Cas9, will you be able to fully reverse not just the sort of things at the level of the neuron, but also the behaviors that they've accumulated over the years as a result of autism spectrum? Again, it's really controversial for me. I'm hoping to share that paper. It is a CRISPR paper that we'll be looking at down the road. So when we're looking at CRISPR, and we looked at this earlier on, this is sort of the generic structure that you'll often see. This is the nuclease itself. So this is something that we would have to engineer. We don't have this nuclease within our genome. We don't have it in the mammalian genome. It is a bacterial nuclease, and so we would have to engineer this. We would have to take its sequence and deliver it to the mammalian genome. But if we were able to do so, this is sort of the nuclease that we would see, and it does cut on 2 different strands of DNA, just like FOC1. These are not dimers though. If we were to take a look at the nuclease activity, within Cas9, we have 2 different nuclease sites. So we have one which is known as HNH, and we have another one which is known as RUBC. And these 2 will cut on a directed area of the DNA that we want to target, that we want to actually be able to cut, either because we want to get rid of something or we want to introduce a repair at this site. Whatever that reason is, we would use these 2 different nuclease sites on this Cas9 molecule. And in addition to that, we have to target it just like we did with talins, or just like we did with the zinc finger nuclease modules. We have to tailor it so that it's only going to a specific region of the DNA. It would be useless to just randomly chop up DNA. And so we need to generate what is known as this guide RNA, often denoted as this SG RNA. And this is part of a larger complex, as shown on the top right. That's also part of this part in orange. So the guide RNA is specific to the genome sequence, and the rest of it contains tracker cRNA as well. So we have to take this part, which allows it to sit within the Cas9 nuclease, and then this part targets specific regions of DNA, and this guide RNA is what we're going to be designing in the next lecture. How do we target something in Parkinson's, or how do we target something in Alzheimer's disease if we think that that will help? We'll also see that oftentimes in the papers, starting today, when we are introducing this nuclease into the mammalian system, we need to see that it's actually being expressed. So we often tag it with the GFP molecule so that we can see it fluorescing. And we know that the cells have actually taken up not just the GFP, but have also taken up the nuclease as well. So again, in a little bit more detail, when we look at these catalytic nuclease domains, we have this RUBC domain and we have this HNH domain. Each of those is targeting a specific strand of DNA, just like FOC1. It forms a dimer, and it will cut, one of the FOC1s will cut the individual strands of DNA. And again, we know that we have to target it to this specific region using a guide RNA, or SG RNA, which is part of a larger complex, the CRISPR RNA and the transacting or tracker RNA that I showed you earlier. And this is something that we don't need to necessarily design. It's something that we use sort of as a template. Any questions on that or anything that you need to know? So you need to know that the CRISPR, the Cas9 system is indeed a nuclease. It does have two different nuclease catalytic sites, the HNH and the RUBC. There are a few more details that I'm going to point out to you here about the nuclease site as well. There is this site-specific cleavage of the DNA that we want to target. We want to cut something on a particular sequence. We use that guide RNA that I've been mentioning to you as well. The other thing to make note of is we will have what is known as a PAM sequence. And this PAM sequence is this protospacer adjacent motif. That is what PAM stands for, the protospacer. The A is the adjacent, the motif. This is on the opposite strand. So we have this guide RNA, and if I go back, we have this guide RNA here. This is what we're targeting. The opposite strand, which is what we've been looking at, has this PAM sequence that is there. This is often associated with the RUBC site or the RUBC catalytic domain. Okay, so again, keeping that clear, I can tell you last year when I asked a very specific question related to the nuclease site and how we would know if we're using one site versus the other, knowing this diagram and what I've just explained to you about where the PAM sequence is located is important. And I do want to emphasize it's an application-based term test this year. I'll give you some more examples in this week's study guide, but please make sure that you know what this PAM sequence is. And this PAM sequence, as denoted here, usually has this structure. Those of you who are in genome biology or other genetic courses, how long does this PAM sequence normally, how long is it? So here it's denoted as 3 nucleotides. Is it always 3 nucleotides? Can it be longer? So usually this PAM sequence is about 3 to 5 nucleotides long. The shortest that it can be is sort of 3 nucleotides, which is often what we see when you're designing it. You'll often use 3 nucleotides, but it can be as long as 5. And when I mention things like NGG, so 5' to 3', the sequence for this PAM sequence is NGG. What does the N stand for? Again, just making sure we're all on the same page. It does not stand for neuroscience, so if you're thinking neuroscience, it's not it. Nicotine is not right either. Let's think like biologists and not like neuroscientists, just for a second. Nucleotide? Yeah, or any nucleotide, right? So it could be like anything. It could be A, C, G, or T, but here we're looking at any of these nucleotides. So when you see NGG, it just means that you have to have those guanines, but you can have anything before that. So there are some real advantages and disadvantages to CRISPR-Cas9. These are still true, even though we've been looking at this since about 2013. Overall, these trends are still pretty much the same. So one of the things that we know that if we're looking at talons in B or we're looking at zinc finger modules down here in C, it's much more complicated for me to be able to design and deliver these enzyme and talon and zinc finger nuclease complexes to a cell. It is much more work. It's much more challenging to be able to do this because I have to do this for 2 separate strands, right? So I have to design a FOC1 construct with the enzyme, and then I have to design these talon modules, and that's on one strand. And then on the other strand, I have to design something completely different, and they have to align and they have to bind exactly. So I have to design it twice, and both of these have to work because we need this dimerized complex. That's challenging, and I have to deliver these 2 different constructs. Here, I only have to design this one guide RNA, and I also have to get the Cas9 in, but that's it. I don't need to worry about having to design 2 separate modules, which may or may not work, and if they don't work, I have to go back and redesign everything. The other big advantage that is really the strength of CRISPR-Cas9, if I wanted to attack more than one region or modify more than one region of the genome using TALENs or the zinc finger modules, I actually have to design like, double the amount. For CRISPR-Cas9, I can attack multiple regions simply by having more guide RNAs. I don't have to like double everything up. I don't have to have like an excess number of these FOC1. I should be really careful how I pronounce FOC1. These different nucleases, and I think when you're looking at this, the design is much more elegant and much more simple. I can use the same Cas9 nuclease and simply have different guide RNAs to target different regions. The nuclease will be there all the time. They don't have to be associated with the modules that you have here. So it's a much more elegant system. And what would be the advantage, though, if everything worked? What would be the advantage of this system, BNC, where we're using zinc finger nucleases versus the Cas9 system? So these are more complex. I have to do double the amount of designing, hoping that everything works. But if it did work, what would be the big advantage? Yes? Less off target. So I've designed two different types of modules to target one specific region. If they're both working, I know that it's going to target that one region. The guide RNAs, there could be some off targets. I actually wanted to target this sequence of DNA, but because of its similarity to another sequence, it could actually be targeting a different region. So there are less off targets with BNC, but the big advantage is that I can do multiplexing, attack multiple sequences at the same time, or do editing of a stretch of DNA using CRISPR-Cas9. And it is a very big advantage for us. All of that sort of, again, emphasized for you on this particular slide. So one of the big things to make note of right now, it's still a limitation, although people are working on workarounds to not necessarily have to take into account this PAM sequence, but in order for us to design this, we need to be aware of that protospacer adjacent motif sequence. Again, it's only a stretch of three nucleotides, but we are kind of limited by having a guanine at one of those positions. So again, it's one of those restrictions that we have right now. So CRISPR-Cas9 doesn't work on every single sequence that we want all the time, but we hope to be able to get past this sort of limitation of having that G at that 5' end. And again, there are less off targets than CRISPR-Cas9, although the efficiency of CRISPR-Cas9 is always improving. It's also very, very controversial. For those of you who have taken other genomics or genetics courses, what is sort of the biggest controversy around the off-target effects of CRISPR-Cas9? Big controversy, it was kind of reversed in the summer, maybe, yes, Tom? Germline, potentially. Maybe not there at that stage yet. Anything else? So there was a massive paper that came out that was actually retracted about a year and a half ago, saying that if you were using CRISPR-Cas9, you were able to detect a number of different off-target effects throughout the genome. That paper has since been overturned because they didn't do the proper control. Somehow it got published. It was an N of 3, and somehow that got published. It made all the news. People stopped dropping the CRISPR-Cas9, and then follow-up papers have actually shown that, in fact, the number of off-target effects are becoming more and more minimalized and minimized in terms of targeting the specific region. So I think at this point, it's fairly safe to say that CRISPR-Cas9 is going to increase in terms of its specificity, even without having this heterodimeric construction. I create two different things targeting one specific sequence that makes it much more specific, as I mentioned, but CRISPR-Cas9 is rapidly catching up to this. Currently, this limitation that you see here is probably going to no longer be as relevant. So some of you who are taking fourth year courses will now know that mitochondrial DNA is also now being targeted by CRISPR-Cas9. It used to be thought, because some genes were encoded by mitochondrial DNA, not necessarily genomic DNA, that this was off base and we couldn't actually influence that, but there are papers that are coming out targeting mitochondrial DNA as well. So maybe down the road, this last sort of criticism or disadvantage of CRISPR-Cas9 will no longer be as relevant. Again, so I also wanted to mention, because this was shortly after morpholino oligos, people wanted to look at altering function and expression of mRNAs and therefore proteins by using a technique known as RNAi. And RNAi has recently come back in the news because it is one of the first approaches that have been approved by the United States FDA for use in therapeutic trials. So this is beyond sort of phase 3 clinical testing. It's now being used, and RNAi is a technology that I think, because it's been around for about 30 years, will be an important one until CRISPR-Cas9 catches up. So the RNAi or RNA interference is actually used a lot in research. It's still used a lot in research. We use it both in cell lines as well as in animals, in vivo. We can alter RNA expression, and by altering RNA expression, it's directly influencing gene expression. For the most part, RNAi, we have to continually deliver it. So RNAi gets broken down fairly quickly, and so the results are temporary. And it can also, in some cases, be fairly non-specific, and we need to actually go back and revisit that same sequence over and over again by re-delivering that specific construct. Again, one other thing that I'd like you to be aware of, because we're talking about guide RNAs, and these RNAs are indeed RNA. It's a tracker RNA plus the guide RNA that you are creating. This means that they will have three-dimensional hairpin structures, and this means that you have to design them fairly carefully. And again, we also want to, just like with RNAi, also be aware of the fact that sometimes these introducing RNA can also introduce some immune effects. We're not 100% sure if these guide RNAs, because they're relatively short in sequence, will actually activate the immune system. But there is some suggestion coming out in the literature that suggests that maybe we have to be aware of possible immune-mediated effects with the introduction of these guide RNAs as well. So in terms of the CRISPR-Cas9, as we'll go through in lecture next week, and again, we didn't do this last year, so it is more specific to this year, we'll be designing one of these guide RNAs where we'll be looking at how do we make sure that the RNA has about 40 to 80% GC content, so the different nucleotides have to have GC at 40 to 80%. Can't be too long, so it has to be somewhere between 17 to 24 base pairs. It becomes more complicated because it does form these other structures if it gets too long. And again, we'll be looking at the on and off target effects as well. So this part is one we don't have to design, and this is sort of the part that is being highlighted by everything that you see here, the GC content, the length. This is the part that we're going to be designing in the next class, and we'll be looking at all of these other effects today, which is how do we actually get this into an animal? What happens after we get it into an animal? How do we analyze some of those changes that we think we should be observing after introducing the guide RNAs into an animal, and how do we actually deliver it? Any questions on this before we go on, or anything that you want to chat about in general? Oh, okay. Again, you don't have to memorize this figure or this diagram or anything else. There are a couple of key points that I wanted to point out for you as we go along. Most of these are delivered via virus, so we will either inject it into an animal, and I'll show you some examples of that a little bit later on. We will have to engineer this bacterial nuclease, and this is the genetic sequence that we would have. we would have to introduce it into the mammalian cells using some kind of mammalian promoter system. And then we often have the guide RNA, if we can, packaged into the same virus. We don't want to have two separate viruses infecting the same cell, because one virus may infect and you might get the nuclease, but you won't have the guide RNA. So again, it's a consideration. We have to package everything into the same virus, and then we have to have some way to deliver it and then have it targeting the organ system that we need to see any changes in that we want. So one of the things that, again, I'm just going to mention in passing, we tend to use this system. There are a whole host of potential viruses that we can use to deliver this gene that we're constructing. We tend to use an adeno-associated virus, and all of these adeno-associated viruses have some different technical considerations. So some of them are bigger, some of them package things smaller. And we want to make sure that we can package both of them into the same viral vector. We would prefer to stay away from infecting a cell with two different viruses, because they may not work all the time. So we need to address whether or not we have two viruses or we can package it all into one single virus. Okay, so I'm now going to take you through those papers that I mentioned in passing in the last lecture. We'll cover Eric Olson's paper at the very end, or at the very beginning of next lecture, to see what the big change was in terms of the canine model system. But I'm going to take you through this. We're going to see a lot of these types of diagrams. So if you take muscle, so imagine for a second we're looking at the length of skeletal muscle. If I were to turn it this way and then cut it like a baguette, and we're looking at the baguette, around the outside of these different muscle fibers that make up the muscle itself, we would have dystrophin. So the things that you see here in green are all of these muscle fibers together. So if I had a whole bunch of baguettes together in a bundle, actually sounds like a really good lunch right now, right? Close, we're getting close. And I were to cut it in cross-section, you would see the outline, and that would be the dystrophin that is on the outside to link it up with the extracellular matrix and the dystroglycan. When we have Duchenne's muscular dystrophy, those are gone. We don't see those outlines anymore. And then with gene therapy, if we could somehow bring them back, this is what we might actually observe. Some of those muscle fibers would start having the expression of that dystrophin again, which is what you're seeing here at the bottom. So we're going to go back and revisit a couple of things really quickly as we go through these slides, and just to make sure that we're all on the same page, and why we talk about zebrafish and why we talk about MDX mice. There are some advantages and disadvantages to these different systems. Again, we don't need to worry about all of the details here. These are individuals, by the way, for various reasons. They were all interested in this technology. So if we're looking at Charles Gersbach over here on the left, again, names not being important, Amy Wagers here in the middle, and then Eric Olson here, and his paper is the one that we're going to be looking at again next week in canine model systems. They've all been interested in muscle for different reasons. Do any of you know Amy Wagers? Possibly one of the most controversial scientists in the last 10 years. Any ideas? So she's actually been interested in exercise and muscle tissue, and whether or not muscle tissue can be regenerated or rejuvenated. So if you have like young blood versus old blood, she was saying that young blood can actually rejuvenate muscle, and it's been a very controversial thing, which is why she is involved in Duchenne's muscular dystrophy research. So we've already talked about this previously, that Duchenne's muscular dystrophy, X-linked condition, affects boys, 1 to about 3,000 to 1 to about 3,500. So it's fairly common. There is not just muscle, skeletal muscle that's affected, the muscle in the heart is also affected. Heart failure is also very common in Duchenne's muscular dystrophy. And we know that the dystrophin complex including things related to dystrophin like dystroglycan, is also going to be affected in this particular disorder. And if you don't have dystrophin, that it's going to cause muscle degeneration and then eventually the loss of muscle tissue, which is called myopathy. So in the past, trying to get through this is been really, really challenging. The dystrophin protein itself is really large, so trying to get something of similar size into skeletal muscle has been very, very challenging. And then, again, this is one of the reasons why Eric Olson has started using dogs. We need long-lasting dystrophin expression. So the papers I'm going to show you, they've followed the animals for about half a year at most. But we don't know if this is going to be a long-lasting change at the genomic level, which is, again, a big challenge for us as well. Again, for these papers, all of them are going to be using CRISPR, just to make sure that we're on the same page. Because CRISPR works on two different catalytic domains, we have the HMH and the RUB-C. They will make two different cuts on DNA. That means that we're making a double-stranded break. So both strands of DNA are going to be disrupted. And depending on how we set it up, this will either end up being repaired through non-homologous end joining, and we might have had an insertion or a deletion, or if we provide a template, maybe we can actually fix something that's wrong at the region that we have just cut. So we will either have homology-directed repair, or we'll have what is known as non-homologous end joining. Again, we need to go through in these series of papers all the things that I've just mentioned to you, how the CRISPR works, whether or not we have homology-directed repair, if we have a donor DNA, can we fix that region that is mutated? That's one approach. Or can we simply disrupt the function of a gene? So let's say, for example, you are carrying a toxic gene. If you're expressing that gene, we need to get rid of it, or we need to reduce the expression. Can we simply disrupt that gene by introducing either an insertion or a deletion, putting things out of frame, and disrupting that gene function that way? So these two approaches are the most common in CRISPR-Cas9. And in Olson's group, which is the one that I'm concentrating on, he's going to try to correct a single nonsense mutation in the Duchenne's muscular dystrophy gene by homology-directed repair in the germline of these MDX mice. He's done this previously, and in this paper, he's actually doing this not in germline and not trying to correct it at the germline level. He's doing this in mice that already have the disorder. So trying to fix this for someone who already has the disease, much more useful than trying to affect the germline expression, which he has also done before. Okay, so these are all in postnatal muscle tissue. This is going to be using a particular variant of the adeno-associated virus. This is known as AAV9. So again, you can have AAV1, AAV8 is very common, AAV4. AAV9 is very tropic for muscle. Okay, so the virus likes to infect muscle tissue, particularly muscle tissue that have elements of like actin and myosin. So it has tropism, or is tropic, is drawn toward or will target skeletal as well as cardiac muscle tissue. So again, these large structural proteins, like dystrophin, have a lot of different regions that are associated with them. And it turns out that if you look at other studies that have been done, about 80% of the Duchenne's muscular dystrophy mutations, they could actually benefit by having what's known as exon skipping. If we could somehow get beyond that premature stop codon, or the truncation that exists, then they might actually benefit by having this, because 80% of the mutations that are identified with Duchenne's muscular dystrophy have patients that have a premature stop codon that exists. So again, this is a more detailed map of dystrophin. So this is dystrophin here at the top. We've seen this several times already. Again, you don't need to memorize everything that's on this particular slide here. These are color coded for you. So the actin binding domains here are in yellow. This is the protein itself, and these are all of the different exons. So you can see there are a number of different exons down here on the left. In the human version, we have 79 exons that make up the full version of dystrophin. And over here on the right in C, we have the exons from mice that will encode something very similar. So 79 exons in mice, 79 in humans, so very, very similar to what we've seen in both species. And again, just to orient you a little bit, to make sure that we're all sort of on the same page, if you were to look at these different exons, and you were to look at how these would form the mRNA, you'll see that there are some different orientations. So some of these simply look like boxes that you see here. So for example, exon 23, 24, and 25 are boxes. And then we have other ones that look very different. They look like arrows. So for example, exon 12 and exon 11 are not simple boxes. They look a little bit different. These are different splice sites. And these splice sites allow for the mRNA from these exons to interact with each other in an appropriate fashion. So I want you to keep this in mind, for example, if we were to eliminate, for example, exon 25, would exon 24 and exon 26 create an mRNA that would result in a functional protein? They would, because those two would have compatibility with each other. But imagine for a second if I decided that I wanted to eliminate exon 43. So would 42 and 44, would they be compatible with each other? They would not. They would not actually form a functional mRNA, and therefore they would not produce the dystrophin that we see here. So having some of these domains that we can actually not worry so much about actually kind of works to our advantage. And about 80% of the types of Duchenne's muscular dystrophy involve regions where, for example, we might have a mutation that creates a premature stop codon at one of these boxes, allowing us to kind of eliminate them. And again, that's really the only thing I want you to take away from this slide, this figure, is to understand that some of these regions are splice donor and acceptor sites, which are those different arrows, and some of these are not, these simple boxes, which are for things like Duchenne's muscular dystrophy, in things like cystic fibrosis, involving a premature stop codon or truncation. This Adalorn was thought to somehow, when you were given the drug, allow for the ribosomes to skip over the mRNA that had that premature stop codon. It turns out that it's not true, but this is sort of the rationale. If we can somehow get beyond a premature stop codon, even though this drug, Adalorn, doesn't really work this way, or work at all, could we create a fully functional protein by somehow using molecular biology to skip over a premature stop codon that leads to, normally, a dysfunctional or incomplete protein, oftentimes what we see in things like muscular dystrophy. And that's sort of the idea that Eric Olson and others had. Not at the germline level, but in a postnatal mouse that's been born, that carries this potential stop codon, could he use the CRISPR-Cas9 system to actually skip over that premature stop codon that has been generated? So here is this MDX model, and this is the same MDX model. If you could eliminate that the next exon over, 24, could actually rejoin with exon 22, and as a result, he would lose one of those domains, just as we have theorized, but he would be able to create a functional protein, much like Adalorn was suggested to be able to do. So he created sort of this construct that you see here, couple of things to keep in mind. He has two constructs. He's not packaging it into one. He's packaging it into two separate constructs. He is delivering the nuclease, the SP Cas9 system, and he is also delivering in a separate adeno-associated virus. He is delivering the guide RNA, and this guide RNA will also have GFP associated with it as well. So his idea is, can we somehow target exon 23? Can we get rid of exon 23 using CRISPR-Cas9 by eliminating exon 23 and allowing exon 22 to now join with exon 24? Can he restore function by deleting or having a delta version of exon 23? Can he restore function to these mice? Again, it's a really big proposition. And one of the things that, again, I need you to understand, in this MDX mouse model that he is using, he has a number of different model systems. I'm not going to ask you to memorize this. There's a lot to this. I need you to be familiar with all of these different techniques. So one is that he's going to inject the adeno-associated virus and the guide RNA, because you need both. You need to eliminate this, and I'll go through that in some more detail on the next slides. He introduces this either through intraperitoneal or IP injection, and this is done 1 day after the mouse is born, or he will inject it directly into skeletal muscle itself, intramuscular injection here, or retroorbital. How many of you have heard of intramuscular injection before? So some of you have. IP injections, intraperitoneal into the stomach, into the peritoneal fluid. And which of these do you think would target muscle the best?
Intramuscular?
It would make sense that intramuscular, so if there's a problem with the muscle, let's inject the virus there. But he's also trying different routes of delivery as well. The last one, retroorbital. How does that work? What is retroorbital? Relying on your knowledge of Latin and physiology and all that good stuff. Any ideas?
Behind the eyeballs?
Behind the eyeball, right? So which of those would you want to have? Probably none of the above, right? It's one of those times. So again, he does this at different timelines. So he'll do it like 12 days postnatally, or he'll do this 2 weeks after they're born, and then he'll follow them for like 12 weeks at a time. One of the outputs that he looks at is, can you get the expression of dystrophin back? So these holes that you see, all you're seeing is the outline of the muscle fiber, because that's where dystrophin is. In the MDX mouse that has degenerating fibers, you don't see that anymore. They're all gone. And then after intramuscular injection, you'll get sort of the presence of the dystrophin again, sort of at the same time. When you do H&E staining to see if you get any inflammatory cells, etc., when he is injecting into the tibia, the anterior tibia, the muscle, the skeletal muscle there, and then he does histology on it, he sees that there is a reduction, for example, in inflammatory cells, and he sees less of the necrotic fibers, and he sees better numbers of muscle fibers, suggesting that intramuscular delivery of the CRISPR-Cas9 and deleting that premature stop codon after the mouse has been born actually is beneficial to the mice histologically. Again, looking at the GFP, also showing exactly the same thing in terms of the changes that you see there. I'm just going to skip because I can come back to these a little bit later on. In terms of the way in which he delivered these different the different types of viruses, this is what he did. So if you are very, very squeamish, it's a good time to look away now. It's not very pretty. I can tell you I've never done this either, but it's an interesting technique. So this is retroorbital. So you take a syringe, you have it filled with the virus, and you go in right behind the eyeball, and you inject behind the eyeball, not into the eyeball, but behind it. There's a number of blood vessels behind the eye, which is what he's targeting here, and you're done. And then you wait to see if the gene therapy has actually worked. So you might wonder, why do this versus like an intravenous injection, which you're probably more familiar with. So I've done this technique, and I can tell you of the two, I'd actually rather do this than do this. And there's a good reason why. You have to be able to immobilize the mouse. You actually have to find the tail vein in the mouse, which is incredibly challenging. So to get them to do this and then to inject appropriately into the vein, and they're going, if he does it right, the animal will turn blue on its legs, on its feet, as well as on its ears. But it's incredibly challenging. I can tell you that it's not easy. It's also painful for the animal as well. So you can see that the tail is kind of turning blue. You'll see that the feet turn blue because it gets targeted immediately to the different blood vessels. And so will this technique too, by the way. If he had injected a blue dye, you would actually also see the feet turning blue as well. So it may sound like a really bad, terrible technique, but this is actually less stressful for the animal because they're asleep while this is done, and it actually is not as bad as it looks. You guys feel comfortable doing that in your labs? No, I wouldn't either, to be quite honest. Like, this is tough enough, but that's actually challenging too. So I have to apologize, I didn't go through all the slides. This week's study guide, I will take you through just up to what we covered. I will take whatever we haven't covered, put it on to next Monday's class. Next Monday is the last class we have before the term test. Just a reminder, whatever we get through in the next class, we'll finish at 12, we'll take another quick break, and then at 12, from 12:15 to about 12:35, we'll have a review on what you need to know for the upcoming term test. Does that work for everyone? Okay, so I'll see you all next week.